Hey, welcome back. Today we are taking a look at a special class of proteins called enzymes. Alright, so starting off, a little bit of chemistry. The making and breaking of chemical bonds, as we see here in this picture, is called a chemical reaction. So we're starting off with two white molecules that are bonded together and a red molecule that's bonded together. And what's going to happen, this arrow is showing, is that we're going to break these bonds, break those bonds, and we're going to rearrange them so that we have the two white atoms now bonded to the red atom, and down here, same thing, two white atoms bonded to the red atom. And this is hopefully a familiar chemical equation for you. We're adding hydrogen to oxygen to make water. And so we're visualizing this chemical reaction. So that's what we're looking at here, is chemical reaction, breaking and reforming of chemical bonds to make new substances. So when we write these chemical reactions, that arrow indicates the conversion from starting material, which we call reactants, to the ending material, which is called products. In a cell, all the reactions that occur are called metabolism, and that's an old word. It's from the Greek metabole, which means change. So we're changing substances from one thing to another, and that's metabolism. It's all the reactions within a cell. Many reactions involve in metabolism occur spontaneously. So spontaneously means they happen all by themselves. They don't need to add anything, they just happen. Imagine you have a match, it's burning, it's on, and you get a paper a little bit close. That paper is going to go up in flames. That is a spontaneous reaction. However, they don't all get started or move very quickly. They are imperceptible, meaning they don't happen as you can watch them. Think of rust forming on a car. You can watch your car and you won't see the rust form, but given enough time, I'm sure we've all seen old rusty clunkers out on the road, that rust certainly does form. Again, over time it does form. It is spontaneous. It can just be really, really slow. Another example is the hydrolysis. So adding water, hydro, to break, lysis, hydrolysis, we're breaking with water. The hydrolysis of sucrose to form glucose and fructose, it occurs spontaneously. So it will happen no matter what. The problem is sugar water can sit and wait for years before it starts to break down. So it will break down, but it might take years for it to happen. So when you eat sucrose, you need to speed that process up. You can't wait years for the sucrose you had at lunch to dissolve into glucose and fructose. We need those glucose and fructose immediately. And that's where we come in with our catalysts. In order to speed that reaction up, the reaction above, where we went from sucrose to glucose and fructose, we add what we call the enzyme sucrase. And then all of a sudden, the sucrose will be hydrolyzed or be broken up within a matter of seconds. So a catalyst is a chemical reagent that changes the rate of reaction. It can speed up the reaction without being consumed by the reaction. So that's an important concept there. Catalysts don't get used up. They're not reactants, and they're not products. They're on neither side of the arrow. All they do is speed the reaction up. They're not changing the reaction at all, just speeding it up. Enzymes, very specifically, are catalytic proteins. So the class of macromolecule from a couple chapters ago, enzymes are proteins, and they're catalytic, meaning their whole job is to speed up chemical reactions. Very specifically then, so thinking back to that reactant, the left side of the equation, what we started with, when we're speaking about an enzyme, the reactant is called a substrate. What happens in an enzyme catalyzed reaction is the enzyme binds to the substrate, or it could be multiple substrates. You could have more than one substrate. It could be putting two things together. And enzymes, very important, enzymes are substrate specific, meaning they're only going to work with one substrate. And that a lot of times has to do with how it fits. So here we have an enzyme in purple, labeled E, and here we have several different substrates. Well, as you can see by the shape of the active site, that's the area that the enzyme matches up with the substrate. This enzyme is looking for this round substrate. Some of these other substrates won't work. Substrate A might work a little bit, but substrate C is definitely the correct shape to fit, and it's going to work the best. So sucrase, the enzyme we looked at the previous slide, it only acts on sucrose. It's built to fit only sucrose, and it'll reject other disaccharides like maltose. It will not break down maltose. You'll notice this a lot in biology. We name our enzymes very similar to what they act on. Big difference is that enzymes usually end in ASE or IN. So sucrase, there's that ASE ending. That should be the signal to you that you're working with an enzyme. What does it break down? Well, it breaks down sucrose. The word kind of matches. So again, enzymes are proteins, and proteins are macromolecules, and they have very unique three-dimensional shapes. And that's important for all proteins, not just enzymes, but for all proteins. So how specific that enzyme can be is all dependent upon its shape. And the active site is the important part of the shape. So the specific shape of the enzyme, where it interacts with the substrate, is called the active site. We saw that on the previous slide. And the whole ability of the enzyme to be specific is all depending on that active site. If you have a round-shaped active site like these here, you're going to interact with round substrates. If you had a square-shaped active site, you'd interact with square-shaped substrates. If you had one round and one square, you'd interact with something that has one round and one square as a substrate. So the shape is everything when it comes to enzymes. And as we mentioned already, enzymes are neither reactants nor products. They're not used up. They're not a part of this reaction. Ultimately, at the end of the reaction, so here's the reaction taking place. You started with your products. We call them substrates in an enzyme catalyzed reaction. Here are the products. When you finish, the enzyme is exactly the same way it was started. And so it oftentimes goes and works with another substrate. It'll match up with another substrate. And so it can be a cycle where that enzyme is never used up. It just keeps going, keeps making more products out of more substrate. So again, a little bit of review from a couple chapters ago. So proteins have very specific three-dimensional structure, and that structure is sensitive to its environment. So like all proteins, there are conditions under which the protein will work optimal. Generally, those environments are pH and temperature, so how acidic or alkaline, that's basic, something is, or how hot or cold it is. When proteins are not in the right environment, so they're not in the right acidity or in not the right temperature, they will unfold and they'll lose their shape. And that process is called denaturation. So here's a shape. Again, you see the very specific shape that this enzyme has. It's going to fit in with that substrate with a very specific shape. If it becomes denatured, it's going to lose its specific shape. And that's no good. Now it can no longer do its job. It can't fit with its very specific substrate. So again, temperature and pH are the main environmental factors when we're talking about all proteins, very specifically enzymes and enzyme activity. So you're going to see quite a few graphs in this chapter that tell, again, read temperature or pH, and you can tell the optimal temperature by looking at the height of the curve. So most humans, if you think about our body temperature, it's about 37 degrees Celsius. So most of the enzymes work in our body in a range between 35 and 40 degrees Celsius, right where our body keeps them at 37 degrees Celsius. So those would be the black enzymes here, right at the optimal temperature for humans to be at. Bacteria, they can go all over the place. So don't think just because we have this particular strain of bacteria that all bacteria are heat specific. They aren't. Some are like us. In fact, lots of bacteria live within us. Others, their curve might be way back here towards the cold end of the spectrum. So bacteria are, or can be, very different. Here's a thermophile who is heat tolerant, and his optimal temperature for his proteins to work is around 77 degrees. And similar to temperature, this graph took a look at temperature. We can also have optimal pHs. So we've got an example with pepsin, which is a stomach enzyme. It breaks down food in your stomach. And guess what? You have a very acidic stomach, a very low pH. That means very acidic. So that enzyme works best at a low pH. On the other hand, we also have trypsin, which is in your intestine, and that has a much larger pH, a much more basic pH. So go figure, trypsin functions best at a higher pH because it lives in an area where it needs a higher pH. All right, I think that does it for our first lesson in Unit 5, Enzymes.